So good morning, good afternoon, or good evening. So this is the third Meet the Scientists webinar series. As a reminder, the Meet the Scientists webinar hosts teams of investigators working to end Alzheimer's disease and other dementias. And it's an opportunity for everybody that is listening and watching us right now to meet the team and also ask questions about what they're doing and learn more about the exciting research that they are doing. So today we have the pleasure of hosting the Healthy Brain Project team, and we're going to learn about what they are doing and all the exciting work that is ongoing. Before we go to then, I just want to remind that this is something that is intended to be doing monthly. So the next one is going to be in February. So we have the Meet the Manbricks Lab, that is the group from Columbia University in February. In March, we're going to be meeting the UCSF ADRC. In April, the Rush Alzheimer's Disease Center, and in May, the University of Kansas Alzheimer's Disease Center. So for more information and registration, you can go to this website. Now to start, I'm going to pass it to the team, the director of the Healthy Brain Project.
Thank you, Yakil, and thank you for inviting us to present. Hello, everybody. Thanks for staying up late, getting up early to come in and meet us. I am just going to share my screen. So hopefully you guys, I think, see that. Probably. So we're the Healthy Brain Project. It's comprised of Matthew Pase, Rachel Buckley, Nawaf Yassi, and I. So we co-lead the Healthy Brain Project, and we've also invited our postdoc Emily and our PhD students Lisa and Kath to also come and showcase a little bit about their research projects. Just to kind of give you an overview, Healthy Brain Project sort of exists in a bit of an ecosystem. The core academic partners involved in this are Monash, Florey, Melbourne Uni, WeHi, and the Royal Melbourne Hospital. So we're obviously based in Australia, in Melbourne. And we started off at the Florey, and we've recently moved across to Monash. Our core industry partners are Cogstate and Cantab, and that will become evident in a little bit. We collaborate across multiple academic centers, and we also work across a range of different cohorts as well. We're supported by both Dementia Australia and the Alzheimer's Association. So this is just to kind of give you an overview that, we definitely don't work in kind of even though the Healthy Brain Project is a standalone cohort, it was really kind of established in this context, and with the idea that we would be able to harmonize and leverage data across multiple cohorts and multiple groups as well. I'm an associate professor at Monash University, and Rachel and I started the Healthy Brain Project back 4 or 5 years ago now. And it's kind of incredible to see it kind of come this far. So it was established in this whole idea that we want to understand Alzheimer's genesis, and to be able to understand Alzheimer's genesis, we need to start early. So this is data that Beth and I published a couple of years ago now, showing that in amyloid negative older adults who are E4 carriers, they're showing this increase in amyloid over time, and Fagan's group at WashU showing these decreases in a beta 40-40 ratio in those who are E4 carriers, and this decrease is starting already in midlife. And so, it was really based off this idea that Alzheimer's processes are already starting very, very early on, and in order to do that, we need to capture these people much earlier. Now, to be able to do that, we need a lot of people because the effects that we're seeing are going to be quite small, and one way in which to do that really cost effectively is to go online. So the Healthy Brain Project is established as an online study. We recruit people who are age 40 to 70 years old from all across Australia. They completed all of their cognitive assessments and their surveys and things like that remotely and unsupervised. And this just shows sort of the distribution of our participants. Most of them come from Victoria, which is where Melbourne is, obviously, because we did center a lot of our recruitment around there. But it's nice to see a spread of participants all over Australia. If you're not familiar with Australia, it's very common that the middle of Australia is very lightly populated, and the population does center around the coast quite a bit. That said, we do have about a quarter of our participants come from regional and rural Australia, which is really exciting to see, and gives us the opportunity then to also look at things like whether rurality in some ways has any effect on, say, cognition or risk of Alzheimer's disease and things like that in the future. This is our study procedure. So they come online, they consent, they do, they enroll, and then they do a whole bunch of online modules. They don't have to do them all in one sitting. So all in all, it takes about 3.5 hours. They can do they can do it in any order. They can do a bunch at the beginning, and then a bunch a week later. We do recommend that they complete everything in about a 4 to 6 week period. We send them a whole bunch of reminder emails and things like that for them to come back. Once they've completed about 80% of their testing, we send out a saliva sampling kit in the mail, and then they send it back to us, and we collect a whole bunch of saliva on them. And then we do a whole bunch of sub-studies as well, which we'll touch on a little bit later. In adapting tests for, if we're going to do remote online cognitive assessments, then a very big aspect of this is really about adapting cognitive tests for remote assessments, right? So you can't just take your traditional neuropsychological tests and then pop them online and expect them to perform similarly or equivalently. And so we've worked with Cogstate and Cantab to examine how the adaptation of their computerized cognitive batteries would work in a remote and unsupervised setting. This is a paper that we recently published showing that of those who did complete the test, there was high acceptability and also high usability. So people were able to learn the instructions, low error rates. They were able to complete the battery, and that involves just simple tweaks to the design. They seem really simple, but I think they did require quite a bit of thinking through about how do we translate not having a person in there with the participant. So if a person, for example, were to sit in with me in a clinic, I would say, okay, you need to press the K and D keys on the keyboard when the car is red, and tell them when to actually respond during the practice session. Obviously, that doesn't happen in an unsupervised setting, so we needed to actually build those design features into the battery. And then there are other things that we're playing around with, such as these Bayesian models, where we look at algorithms to see if do we even need a practice session, for example? Can we just have them test repeatedly and nominate, say, the first 10 or 20 trials as practice, and depending on the rate at which they learn that, it then rolls into the real trial. So there are all these different things to kind of play with, which are pretty fun. We run a whole bunch of sub-studies. So there's the main Healthy Brain Project. We're up to about 7,000 plus participants now. We're aiming for 10,000. But from there, we draw on our participants to come in to do a biomarker study. So we bring them into the Royal Melbourne Hospital and test them. We collect blood and do a lumbar puncture and all that sort of stuff. We also have a pretty substantial sleep program, which Matt will talk about in a little bit. We also try and understand whether we can detect cognitive changes earlier using high frequency cognitive testing. And then the most recent thing that we're working on is this trial called BetterBrains, which is a person-centered multi-domain sort of lifestyle intervention trial. So the biomarker study is, as I said, conducted in the Royal Melbourne Hospital. It's every 2 years instead of every year. They undergo a full neuropsych battery. They do a lumbar puncture. We collect bloods. We do MR. We also collect stool samples from them, and then we also enroll them into a retinal scan study. So we do hyperspectral retinal scans on them to look at look at whether we can detect, say, amyloid in the retina. And we also then do 2 weeks of actigraphy and then also an overnight sleep study. So it's a pretty substantial battery of tests that we put these participants through. Obviously, it's a much smaller sample. So we're aiming for about 400 participants in this. And, we got to about 90 in 1 year, which was really exciting. But then COVID hit, as I'm sure it's affected everybody. And so we've had to stop. So hoping to restart that very soon. We also do a bunch of high frequency cognitive testing. So this is ORCA, where we tested participants over a series of 6 days. And what this does is it teaches people, it's a paired associate learning task, it's an implicit paired associate learning task. So we teach people the associations between the Chinese characters and the English word, and they learn through just sheer exposure of the frequency of the correct pairs. So we tested participants over 6 days. This is data from ABLE, but we're putting this into the Healthy Brain Project. And what this shows is the modeled effect from the linear mixed model, and figure B is the raw performance scores. The shaded lines and the error bars represent 95% confidence intervals, and we found that over 6 days of testing, the effect size in the rate of learning between amyloid negative and positive individuals was very large, so greater than 2 standard deviations, which is exciting and pretty clinically, I think important. Then we sought to examine learning performance with Alzheimer's biomarkers, and as expected, a higher amyloid levels was associated with slower rate of learning. So for brain volume, we observed an interaction where larger ventricles and smaller hippocampal volume were associated with slower rates of learning, but only in the amyloid positive group. And there was no relationship between brain volume and learning in the amyloid negative group. So the results of this study kind of presents a hypothesis that the cognitive dysfunction in early Alzheimer's disease might be related to this failure to benefit from experience or learn. And so, as I said before, I'm hoping to now challenge this in other samples, including the Healthy Brain Project, and working with Jason Hassenstab at WashU to look at it in his cohorts as well. Now, the other most recent thing that we're doing is BetterBrains, and it really, this figure here shows the overlap of these modifiable risk factors in the Healthy Brain Project cohort, and as you can see, as we would all expect, there's a substantial amount of overlap and shared variance, and so BetterBrains really comes out of this idea that a lot of these modifiable risk factors do share a lot of variance. There is a lot of overlap in the extent to which they affect each other, flow on from each other. So for example, somebody who is obese is probably more likely to also have poor sleep, probably also have pretty poor mood as well. Not always true, but the whole idea is that these modifiable risk factors tend to clump together, and so BetterBrains attempts to try and change it. This idea that you just change one is better than changing none, and even if they start off with changing, say, their mood, that can have flow-on effects onto other parts of their lifestyle as well. So we're hoping to recruit 1,500 participants in the new year. Primary outcome is changing cognitive function at 24 months, and it's person-centered, so it's really driven by that person's goals. So it's not a prescriptive program intervention at all. Before I hand over to Matt, I really just kind of wanted to again show some of the data from the Healthy Brain Project. What we did was looked at, we sampled a whole bunch of people collect a whole bunch of saliva samples from our participants. and you know, genotype them on APOE. We split them into non-carriers, E4 heterozygotes and E4 homozygotes. This figure just shows the magnitude of difference, so you can almost you know, interpret this almost as a standard deviation. The 0 line here represents E4 non-carriers. The light gray bars are E4 heterozygotes and the dark gray bars are E4 homozygotes. So on this side of the figure, these are Healthy Brain Project participants who were assessed on the Cogstate brief battery. We formed attention composites and memory composites, and then we also had subjective a measure of subjective memory as well, which is the ECOG. And what you're seeing here is that, you know, even the average age of these participants are about 52, 53 years old from memory. And, you know, what we're still seeing here is that, you know, this deficit or slight sort of dysfunction in E4 homozygotes. And this is detected in an unsupervised manner. And on this side of the figure, these are able participants, so they're older, obviously, and they're supervised. So all of the able assessments are conducted in clinic and are supervised. They completed the same measures, so the same Cogstate measures. We performed, again, the attention and memory composites, and the subjective memory measure in able was the MAC-Q. And again, we're seeing this sort of paralleled effect in able, which I think nicely kind of demonstrates that, you know, even though we have done all of our assessments in an unsupervised way, we're still seeing these effects of E4 homozygosity in people. And what we ended up doing is pooling the data across Healthy Brain Project and ABLE. So obviously those who are 40 to 50 is all purely HBP, 51 to 60 and 61 to 70 is a combination of HBP and ABLE. And then obviously in those who are 71 and 80 years old, that's just purely ABLE. And what you see is this kind of age effect, that an increasing age effect of, you know, age and APOE for homozygosity on the memory composite, probably because a lot of the E4 homozygotes here are probably amyloid positive. Okay, I'm going to hand it across to Matt.
Thank you, Yan. Thanks to Kill for the invitation to talk today. So I'm an associate professor at the Turner, like Yan. I'm also an associate professor at Harvard University. I'm going to talk today about some of my research interests and the initiatives that I'm involved in. I won't be presenting specific data per se, but sort of just talking through some of my interests, and I'll be happy to chat offline about anything further. So I'm very interested in risk and protective factors for dementia. I sort of come from the understanding that most cases of dementia are mixed, and a sort of strategy to reduce the burden of dementia substantially probably needs to take that into consideration. And I kind of think about dementia much like I think about heart disease, and try and understand its pathophysiology, try and understand its risk factors to see if there are things we can do to reduce dementia burden in the community. So a lot of my research is focused on using large cohorts, and in particular the Healthy Brain Project, to try and understand this further. So two of my main research interests are in sleep and vascular risk factors. Some of our research on sleep, for example, has looked at what aspects of sleep might be most associated with risk of dementia. As some of you might know, there's a huge interest around slow wave sleep because of slow wave sleep's sort of involvement in glymphatic clearance and sort of removing amyloid from the brain. But actually, when we look at our data, we see that REM sleep seems to be most tied with risk of dementia in the future. And those people that have lower amounts of REM sleep actually have a high risk of going on to get dementia up to 19 years into the future. Also interested in vascular risk factors, and we've looked at, for example, adherence to American Heart Association guidelines for preventing cardiovascular disease. And we find that those people that have higher adherence to guidelines have a lower risk of not only stroke and not only vascular dementia, but also a lower risk of going on to get dementia of any kind, as well as dementia due to Alzheimer's disease. People with better cardiovascular health have sort of less cognitive decline, less brain atrophy. And we've also looked at blood pressures, and some of you might know that blood pressures are not sort of distributed evenly throughout the arterial tree, and the blood pressures that you measure in the arm are not necessarily the blood pressures that travel to the brain. And we've looked at the blood pressures that sort of the brain sees by measuring or estimating blood pressures in the aorta that flow up to the brain, and found that these better predict cognitive function as compared to sort of standard ways of measuring blood pressure. We also have an interest in diet, and we've looked at some sugar in the diet and shown that people who have more sugar have sort of more brain atrophy and worse cognitive performance. And on the flip side, people that have better dietary patterns, such as those who adhere more towards a mind diet, have a lower risk of going on to get dementia. In addition to that, we look at other things like environment, and Yen presented data on Healthy Brain Project showing the distribution of people across Australia. And we've looked at even just simple things like where does someone live and how disadvantages to that community. And we're still working on this data, but we're starting to see that even just living in a more disadvantaged community with fewer resources seems to be associated with poorer memory function. Next slide, thanks, Yen. So one of the questions is, so if we know sort of what factors are associated with dementia, can we do anything about it? Can we prevent people from getting dementia? One thing that I'm quite interested in is this notion that risk factors are dynamic across the lifespan, and that sort of, I guess, is twofold. One is that what's predictive of dementia in old age at sort of 40 versus 50 might be slightly different, but then also preventing dementia really does require a lifespan approach. I sort of work closely with Sudha Seshadri and the team at Framingham Heart Study, and that study has follow-up on participants for half a century. And when we take people who are aged, say, 80 or 90, and we look at their brains, on MRI, and then we also do autopsy, and we look at what factors in that person's life best predict their brain health in old age, we find that often it's things that these people did in their 40s and 50s that most strongly predict what their brains look like when they die or when they're very old, suggesting that a dementia prevention strategy needs to be sort of focused on the lifespan and not just targeted to people who are quite elderly. And this fits with the model of Alzheimer's disease having a very long preclinical phase. And so I think one of the challenges is, how do you get people who are younger to care about their brain health and take ownership of their health? Thanks, Jan. So another main interest is on dementia prediction and sort of what factors can we use to predict risk of dementia. So if we know someone's demographics, we know their genetics, we know something about their clinical status, such as their blood pressure or their BMI. What other variables might be informative to know to predict outcomes? So one interest I have is in blood biomarkers, and as you mentioned, biomarker cohort, we're collecting blood as well as CSF, and I'm quite interested in markers of inflammation to see whether different things in the blood, such as markers of neural inflammation, could predict who's at greater risk of declining more fast clinically. And in addition to that, I'm also interested in sleep biomarkers. So I mentioned that some aspects of sleep might be risk factors for dementia, but then also other aspects of sleep might be biomarkers of something going wrong. The sleep-wake centers in the brain atrophy as dementia progresses, and there might be certain cues that you can pick up by doing sleep studies that suggest that someone's at a higher risk, that someone's on that pathway towards dementia. Thanks, Jan. And so in terms of initiatives, obviously there's a Healthy Brain Project. Together with Chandra Himali at University of Texas, I co-lead the NIA-funded Sleep and Dementia Consortium. We're essentially taking hundreds of sleep biomarkers that have been derived from polysomnography and trying to condense that down to come up with signals that are best associated with risk of dementia. In addition to that, I work closely with the Framingham Heart Study, where I did my postdoc, in particular Sudha, who I mentioned, as well as Chandra and Alexa Beiser. And I also lead the International Stroke Genetic Consortium's Cognitive Working Group, where we look at essentially risk factors for vascular cognitive impairment. So within our lab, we're looking for a postdoc. So if this sounds interesting to you, please email us. Thank you.
Hi, everyone. My name is Rachel Buckley. I'm faculty at Massachusetts General Hospital, although I am very strongly entrenched in this group. I did my PhD at the University of Melbourne and still have my affiliations with the University of Melbourne and the Florey Institutes. And now with the transition to Monash University, I'm also a proud affiliate of Monash University as well. So if we look at the next slide, I'd like to just give a very brief rundown of what I do in the US and how that sort of intrinsically dovetails with the Healthy Brain Project initiatives and ideals. So my area of research is in sex differences for Alzheimer's disease, particularly very, very early on in the preclinical stages of the disease, when people are largely healthy, but they do have markers of usually amyloid and tau is what we tend to look at. In the Harvard Aging Brain Study, we look very much at neuroimaging markers. PET imaging is our strength. And so what I have been doing while based at Massachusetts General Hospital is looking at sex differences between self-report of male-female in tau PET and amyloid PET. But I don't just focus on Harvard Aging Brain Study data, just like the other leaders of the Healthy Brain Project. I actually work across lots of different data sets. I look at the Harvard Aging Brain Study, but I also look at the ADNI cohort and quite a few other cohorts, Framingham, RAP, which is the Wisconsin Registry Aging Prevention cohort, Alzheimer's Prevention cohort, as well as the ABLE cohort, which is the Australian equivalent of a lot of these studies. And what I am very interested in is trying to understand why females might show higher levels of tau than males at this early stage. And if we go to the next slide, we show in quite a few studies now, and it's not just my work, but a lot of different cohorts show that women have higher levels of tau than men for after adjusting for quite a few different things. And what we try to understand now is why that might be. And how the Healthy Brain Project helps me with try to understand that question is by looking earlier. A lot of the work that has been done so far has been in older adults, people who are over the age of, or approximately at the age of 75. But there's more and more evidence starting to show that maybe it might be at the earlier stages in menopause, even when risk that diverges between men and women might be starting to occur. So what we gather in the Healthy Brain Project is information on menopause. And it's not just symptoms of menopause, but also the age at which menopause may have occurred. And we also gather information on surgery, past surgery history, and also medication use involving hormone therapy, things like that. So this is just an example of data that we collect. We gather information about symptomatology of menopause, how severe symptoms might be. And you can see across lots of different symptoms on the left panel across the age range. There's actually a lot of stability in some of these measures. So anxiety and panic can be elevated across the age range, but some things that are very, very intrinsic to menopause, hot flushes, sweating, things like that, they do seem to be more age associated around the time of menopause. The other thing that we're starting to look into, this isn't published data yet on the right-hand side, is how these menopause symptoms might be associated with cognition and how this might be impacted at certain ages. We know that age is intrinsically associated with a lot of changes in cognition, and so trying to pull apart the difference between the inflection point of menopause and the natural age-related cognitive decline is something that we're going to try to be teasing apart over the next while. On the next slide, changing tack entirely, I wanted to talk about the passion project close to my heart and very much close to Yen's heart as well. And this is part of the reason why we started the Healthy Brain Project in the beginning, was to look at high frequency cognitive testing, smartphone testing, online digital technologies. These are the ways of the future, particularly since the pandemic has started. We've really noticed that digital technologies is where it's going to be at for testing people. And this is not just to get access to geographically isolated or physically isolated individuals who need access to testing, but also because it's just maybe a safer way, particularly during pandemics. For us, what we're interested in doing, not just for smartphone, but high frequency testing is helpful in terms of building more reliable measures, hoping that people come in once a year, and that time that they come in, they're not hungry or tired or distracted in some way, which might cause their testing to be slightly compromised. If we can do high frequency testing within a short time frame, can we get a better understanding of how somebody is learning or how somebody is performing at that particular point in time? And so we have been in partnership with Jason Hasenstab at Washington University, St. Louis, using his smartphone app called ARC, Ambulatory Research and Cognition app, which please do feel free to contact Dr. Hasenstab if you have more questions about this particular app. But we have worked very closely with their group to implement this this particular smartphone app to see how well it can perform in comparison to other smartphone or other digital technologies in terms of improving reliability for cognitive performance estimation. So moving to the next slide. This is some of the data coming out from the smartphone app collection. This is Dr. Hasenstab's work, but we foresee very similar results in our own work, but just at a larger scale because we have so many people in our study. This is fantastic work from Dr. Hasenstab's group, showing just even in a small group of 30 people, these really quite close relationships between the ARC composite, cognitive composite, and CSF total tau. And as Yen had mentioned, we have the biomarker study. People in the biomarker study, a lot of them are doing the ARC high frequency testing, and we're going to be looking into that data as we get further into the study. And with that, I will be moving to the next slide and handing over to Dr. Nawaf Yassi. Thank you, Nawaf.
Thanks very much, Rachel, and hello, everyone. So I'm Nawaf Yassi. I look a little bit different to that photo. It needs to be updated, obviously. I'm a neurologist at the Royal Melbourne Hospital, and I have a clinical interest in both stroke, acute stroke, and stroke recovery, and also in dementia. And naturally, part of my interest in dementia is really on the overlap between stroke and cerebrovascular disease in general and dementia. And I also work at the Walter and Eliza Hall Institute of Medical Research, or WEHI, where I jointly lead a clinical research laboratory with my colleague, Associate Professor Rosie Watson, who's a geriatrician, and our lab at WEHI is interested in understanding comorbidity in dementia and in aging, and we supervise a number of different clinicians from different specialties, including neurology, geriatrics, psychiatry. And as I said, one of my primary interests has really been in understanding the overlap between different causes of dementia, and particularly one that I want to talk about today is the overlap between cerebrovascular disease and neurodegenerative diseases, or specifically Alzheimer's disease. And so a couple of years ago, I used some traditional MRI imaging, largely structural imaging with T1 and T2 and FLAIR, to try to derive a vascular score that was ready to be implemented in a clinical setting, rather than necessarily being largely a research-based score. And so this score combined a number of different metrics, including white matter hyperintensities, as you can see here in the top right, but also silent brain infarcts. You can see here a posterior cerebral artery territory infarct on the top left, lacunar infarcts, smaller cortical infarcts, thanks, Ian. And in the middle figure here, an entity that's really up until now being just in the space of neuropathology, but that we can now visualize on clinical T1 scans, which is cortical cerebral microinfarcts. And so using a combination of these different markers, we try to derive a simple score that indicated people or participants or patients with high cerebrovascular burden. Next slide, please, Yen. And so in a couple of different analyses, we wanted to firstly look at how the overlap between cerebrovascular disease and markers of Alzheimer's disease varied throughout life. And so you can see here on the left a representation of how this grouping of A beta positive or negative and vascular positive or negative changes throughout the lifespan between the ages of 60 and 90, both in E4 positives and in E4 carriers and in non-carriers. And so, for example, in the top left corner, you see that most people, particularly if they're non-E4 carriers, start off at the age of 60 not having significant amounts of either of the conditions. Now, this is a little bit simplistic, obviously, because it is a thresholded value, but I think it tells an interesting story. And then if you fast forward to the bottom right, you see that that as people age, particularly if they're E4 carriers, this prevalence of overlap between the two conditions becomes the norm, really. The sort of single morbidity picture is a lot more complex, but as you can see, it's less common as people get older, and the comorbidity of the two conditions is much more common, as we would expect, and as Matt and others alluded to. The other thing we wanted to look at is whether this comorbidity had any clinical meaningfulness, or had any effect or association with cognitive performance or brain atrophy. And again, in an analysis from the ABLE study, you can see here that this red bar on longitudinal follow-up up to 90 months showed that the group that had both A beta and significant cerebrovascular disease really declined most out of any of the other groups, both in terms of cognitive performance and in terms of hippocampal volume. Thanks, Yen. Next slide. So in terms of the work in this space in the Healthy Brain Project, as Yen mentioned, we are collecting MRIs as part of the biomarker sub-study, and this will be something that we will look at in the biomarker sub-study. But in the meantime, we have been looking at markers of cerebrovascular risk rather than measurable markers necessarily of vascular insult or burden, and we're able to do that with a large sample size in the overall study. And so if you look at the, this is a figure from our baseline publication earlier last year that shows that although these are largely healthy and cognitively normal middle-aged people, there is a not insignificant representation of different medical comorbidities and particularly cardiovascular risk factors that's not too dissimilar to the Australian community at large. And so you see that we do have a number of people who have hypertension, who have dyslipidemia, who have diabetes, who smoke, who are overweight. And so we've been interested in looking at the influence or the association between cardiovascular risk status and cognitive performance using a remote assessment algorithm. And so in the sort of top Venn diagram, you can see again, as Yen alluded to, this overlap between different vascular risk factors. So these don't occur in silos, but there's a really interesting pattern of overlap, with most people having more than one risk factor. And in a sub-sample of about 2,500 people from one of our first data freezers, you can see that a greater number of cardiovascular risk factors was associated with reduced performance, particularly on the memory composite. And this was the case after adjusting for things like age and education and depression and anxiety symptoms. And this is still work that's ongoing. And with that, I will hand over. Thank you.
So my name's Kath. I'm a PhD candidate at the University of Melbourne, and I'm being supervised by Matt and also working with the Healthy Brain Project as part of my PhD research. So I've got an interest in neuropsychology and dementia, particularly Alzheimer's disease dementia. And I'm really interested in the early detection of Alzheimer's disease and also looking at modifiable risk factors. So one proposed risk factor that needs some more investigation is psychological stress. So it's thought that chronic stress in adulthood may be associated with an increased risk of dementia later in life. And this is the basis to my PhD research. So as part of my PhD, I've just completed a systematic review and meta-analysis demonstrating that aspects of stress are in fact significantly associated with an increased risk of dementia, including Alzheimer's disease dementia. And in this study, we looked at different aspects of stress, particularly perceived stress, stressful life events, and also neuroticism. So through the Healthy Brain Project, I'm hoping to explore the associations of these different aspects of stress with both cognition as well as Alzheimer's disease biomarkers. So I'll be primarily focused on levels of perceived stress, exposure to daily hassles, which are frequently occurring minor stresses rather than major life events, as well as neuroticism levels, which are thought to influence our proneness to experiencing distress in response to stresses. So to explore the associations of stress with cognition, I'll be utilising the online main sample of the Healthy Brain Project. And as Yen already mentioned, participants complete a range of online questionnaires, including measures of stress, such as the perceived stress scale. And as part of this, participants also complete the 10-item Connor-Davidson resilience scale. And so I plan to also explore whether an individual's resilience levels affects any relationship between stress and cognition. And then using the biomarker subsample of the Healthy Brain Project, I hope to explore the associations of the different aspects of stress with Alzheimer's disease biomarkers, including cerebrospinal fluid levels of tau and amyloid beta, which we get from the lumbar puncture, and hippocampal volume from the MRIs. So in this sub-study, I also plan on looking at cortisol levels as a measure of stress, and we can do this using the blood samples that we obtain through the biomarker sub-study. So overall, it's hoped that the results will provide an important step towards viewing stress as a potential modifiable risk factor of Alzheimer's disease. So I'll hand over now to our postdoc, Emily.
Thank you very much, Kath. It is brilliant to be here. So my name is Emily Rosenich and I'm a postdoctoral research fellow here at Monash University. Next slide, please, Yen. So my research interests really centre around better understanding both non-modifiable and modifiable risk factors and their link to multiple outcomes. So neurodegenerative processes, cognitive decline, and dementia. So a lot of us here today will know that there is, these vascular contributions to dementia and now these genetic contributions to dementia that have been increasingly recognised over the past 2 decades. However, the pathways through which both vascular and Alzheimer's disease pathologies actually occur in parallel to kind of interact to kind of drive neurodegeneration, cerebrovascular disease, cognitive decline, and dementia largely remain unknown and need further investigation. So just presenting here today a bit more of a general overview of my research aims and interests. So there are really 2 main aims to my research projects. So the first of which is to clarify how genetic and vascular risk factors interact or, occur in parallel on cerebrovascular disease, neurodegeneration, cognitive decline, and then either progression to vascular dementia or Alzheimer's disease dementia. The second main aim of my research project, and something that I'm really excited about, is exploring the effects of whether we can reduce vascular risk factors in midlife, which seems to be a critical life stage where these vascular risk factors really seem to emerge, and whether that has an effect on preventing cognitive decline in people who are at high risk. So people who are APOE4 carriers, but are looking and comparing this with people who are non-carriers as well, doing this in the BetterBrains trial, which Yen discussed a little bit earlier today, which is a large randomized controlled trial focusing on 4 modifiable risk factor clusters to try and prevent cognitive decline in midlife. So we think that this is really important to explore, because if we have a better understanding of how these risk factors, both non-modifiable and modifiable, are actually interacting, this might help us provide some interesting or important information regarding disease mechanism, and also provide important benefits or information about how we can intervene early, which we know is important, and how we can develop novel disease modifying therapies and strategies to try and prevent dementia. So that's just a bit of a summary of my research interests and aims within both the Healthy Brain Project, as well as leveraging data across multiple cohort studies, again, as Yen alluded to earlier in the presentation. So leveraging data from the ADNI cohort, ABLE, as well as NAC. So thank you very much for the opportunity, and I'll pass on to the next speaker.
Hi everyone, I'm Lisa and I'm a first year PhD student with the Healthy Brain Project team. My research interests are really anything to do with modifiable risk factors for Alzheimer's disease and dementia. Factors that we could potentially target or intervene with to either reduce the risk or delay the progression of dementia, and essentially how the way we live our lives affects our brains and cognition. Next slide, please, Yen. Thanks. So the premise for my PhD research has been touched on a little bit by Yen and Nawaf before, but that modifiable risk factors for cognitive decline and dementia, such as physical activity, low education or depression, have really only been studied in isolation, even though these risk factors may cluster together and share a significant amount of common variance. And as we all know, risk for dementia is extremely complex. So by only studying one modifiable risk factor, we may be getting an inaccurate and incomplete representation of the influence of modifiable risk factors on cognition and dementia. I'll be using Healthy Brain Project data from the online study, from the Biomarker study, and also leveraging other cohorts. And the aim of my research is to study the combined effects of key modifiable risk factors from multiple domains of life on cognition, brain volume, and risk of incident dementia in cognitively normal adults. And we also plan to determine whether the presence of genetic risk factors such as APOE4 modifies any of these relationships. Just to show you a bit of work that we've already done, using data from the main online study, we've already looked into 4 factors of cognitive engagement. So these include educational attainment, occupational complexity, social engagement, and cognitively stimulating leisure activities. And we looked at their combined relationship with cognitive outcomes such as attention and memory and subjective cognition. And you can see the results in this figure on the right here. And we observed that participants who scored highly in 2 or more of these cognitive engagement factors had moderately better memory performance than participants who scored highly in 0 areas of cognitive engagement. And so those asterisks there indicate significant Cohen's d effect sizes. And these results suggested a cumulative relationship between high cognitive engagement across several areas of life and better memory performance. And our next steps will be to incorporate a broader range of modifiable risk factors from different domains, such as lifestyle, so that could be physical inactivity, poor sleep, poor diet, psychosocial, so depression or stress, vascular risk factors such as hypertension, high cholesterol, as well as these cognitive engagement factors as well. And by taking this more holistic approach, we hope that this research will contribute to a more comprehensive and complete understanding of the relationship between modifiable risk factors with cognition, brain volume, and risk of dementia. And we also hope that this research will inform the design of multi-domain lifestyle intervention trials that aim to delay or prevent dementia. Thanks very much.
Thanks, everyone. That's us. This is the Healthy Brain Project team. We have a pretty big group of research assistants who work very, very hard behind the scenes to help us with all sorts of things. Our key collaborators, and this is just all of our sort of funding support. So very happy to take questions from the group. Follow us on Twitter, email us if you have any questions. Visit us if you have any ideas for collaboration or anything like that. We'd love to chat as well.
Thank you so much. That was super interesting. And first of all, I would like to congratulate you because you have accomplished so much that, it's kind of like when you were presenting, I was like, oh my God, it's amazing. So congratulations for all the hard work. I think that especially now in the times of post-COVID, I think this kind of is going to be super relevant. So congratulations for all of that. You had one question in the QA, and I think that is Dr. Lin. So.
Yes, so the questions about potential interactions between A-beta positivity and cognitive performance in the homozygotes in those 10-year age bands. Yes, this is definitely something that we'd like to look at. We haven't done the analysis for A-beta in the Healthy Brain Project biomarker sample yet, so we will look at this interaction. But in the ABLE group, we have looked at interactions between A-beta and APOE4, and absolutely there is an interaction there. So those who are amyloid positive and E4 carriers are also more likely to show cognitive impairment, but also cognitive decline over time. And then to the second question, which is how many cognitively unimpaired homozygotes were those in the late ages? They're all cognitively unimpaired. So we only took, and it was cross-sectional as well. So it's cognitively unimpaired, cross-sectional performance in ABLE.
Great, thank you. There's another question in the chat.
Is there another question?
In the chat, not
on
in
the use of commercial grade wearable devices to collect high frequency behavioral biometric data.
Oh.
Chat instead of QA.
Oh yeah. Yeah, we haven't explored the use of commercial grade wearable devices. We've been kind of exploring different types of methods of collecting data. We've been talking to a couple of people interested in passive data collection. So for example, typing cadence and things like that, but also speech biomarkers. We haven't really implemented those as yet. I think, we'll take, as we enter sort of our 5th year of the Healthy Brain Project, it's time for us to sort of take stock of what we've done, what works, what doesn't work, and then look at sort of new directions to challenge or grow the study.
We're always open for interesting and new ideas, so do feel free to approach us as well.
We do actigraphy, so we do activity monitoring with commercial variables to look at primarily sleep-wake patterns in a subset of the country.
So we have a question about subjective cognition in HBP. Rachel, do you want to take this?
Yeah, sure. So the subjective cognitive measure we use is actually very aligned with the measure that's employed in both the anti-amyloid A4 trial and Harvard Aging Brain Study. It's the cognitive functioning interview, which was initially developed. I actually can't remember exactly where it was developed early on, but was configured and updated by Rebecca Amarillo and others, and has been published in Harvard Aging Brain Study. And then we just changed a bit of the wording in order to be more in line with middle-aged cohorts. So it's a very similar measure to the cognitive functioning interview.
And another one for Rachel. Any information on the type of estrogen that tends to be given in some of these estrogen replacement studies that may not necessarily be the one we produce naturally?
Yeah. That's very true. So we have information, as I said, medication data on who might be taking hormone therapy. We don't go to the level of detail of what exactly the form of hormone therapy it would be. And that's actually very different, sort of problematic, because actually in America, at least for a few decades, it was a very common hormone therapy that was administered, whereas in Australia, I think it might be a little bit more diverse. Although I would refer to Dr. Nawaf Yassi, who is actually an MD who could probably speak to this better, and we would have to look into that further. In terms of the estrogen replacement studies, I couldn't speak to that directly. We don't run those in this particular study, but would be happy to talk offline. Feel free to email me or tweet me at any point. I'm more than happy to speak to you further.
Great.
Okay. Anybody has any other questions? We have 1 minute, so I think that we so thank you again, everybody, for the presentations. I think they were great to hear everything that you were doing. And again, the website is there, the email and Twitter, and this recorder is going to be available. And I'm going to send the link to everybody that registered for the presentation. So if you want to watch it again and learn more about their work, you can just play it again as many times as you want. So thank you so much, everybody, and have a good day. Thank you for
actually. I think there is another question. Sorry,
one more question. Sorry.
This is actually a really cool question, and I think Matt might want to speak a little bit about some of the things we've been discussing a while ago.
Sure, thanks. Yeah, so we did do some work around COVID and the implications or effect that might have on our cohort. So we administered a survey to try and gauge how people were affected by COVID and the effects it had on their mental health in terms of their mood, their stress. And we collected a whole range of demographic data around their living arrangements, whether they live with someone else or by themselves. And we are currently working on the data, so we just sort of extracted it from the website and are looking at it. And I think one of the key things about the Healthy Brain Project is, we have follow-up data on these people, so we can compare how they're presenting during a period of lockdown. And so Melbourne went through one of the harshest lockdowns worldwide, where we were in lockdown for 112 days. And so we can actually take someone's scores on measures of mood during that lockdown period and compare that to how they were feeling 12 months ago, and look at whether there were certain factors that might predict whether someone had better outcomes or worse outcomes during a lockdown period, whether having more resilience or more social supports might sort of affect that and moderate that relationship. So hopefully we'll have more to say on this soon, but it's certainly something we are all looking at.
Yeah, and it's just super interesting because comparatively, Australia has quite a few cases. And in fact, in our Healthy Brain Project cohort, of those who we sampled, we really only have 2 COVID positive cases. So we really don't have that much of an opportunity to understand COVID on, say, cognition and how that develops over a lifetime. But understanding the effects of isolation on the other hand would be super interesting, and that's something that we plan to delve into.
Just as a future collaboration, so we were doing a similar study with the isolation and COVID in Latin America. And we collected data for 11 countries actually from Latin America. So it will be interesting at some point in the future even to compare.
Yeah, that would be really cool. Definitely.
I love this. I love the fact that we can collaborate now. That's great.
Yeah.
Okay,
absolutely.
Any other questions? Check. Any questions? Okay, no more questions.
Well, I just think that the potential for collaboration is just huge because even though, say, America and Australia, we all speak English and all that. So there are a lot of things that are quite similar across the 2 countries, but there are also a lot of very key things, particularly around health, that are quite different as well. And so looking at the differences in how health and Alzheimer's risk then presents in these 2 different groups would be super cool.
Yeah.
I agree. I think policies around how things were rolled out, how things were, how public policy was conducted around COVID will be fascinating to compare between the Americas and Australia.
Okay, so thank you again.
Thank you, Raquel. Thanks for the invitation.
For all of you, so have a good day. For the ones over here, have a good evening. And we will be in touch. Thank you so much.
Thanks for inviting us.
Awesome.
Raquel.
Thanks, everyone.
Thank you.